Why are some amino acids classified as non-polar aliphatic amino acids while some are polar but uncharged? What is the indole ring in tryptophan or imidazole ring in histidine? What does it actually mean? In this video, let us understand the classification of amino acids based on the properties of their side chains or R groups. The first class is non-polar aliphatic amino acids. First, let me explain the term with the simplest amino acid as an example. So this is glycine and the second one is alanine. Glycine is the simplest amino acid. As you see, the side chain is a hydrogen. It's non-polar. So aliphatic amino acids have side chains that consist primarily of carbon and hydrogen, as you see in alanine, forming open chain structures rather than aromatic rings, making it non-polar and hydrophobic. That means these amino acids do not interact favorably with water, forming hydrogen bonds. The next in the list is valine, which has a branch chain structure, a longer branch chain structure compared to alanine. This is also non-polar. The next one is leucine. Leucine is similar to valine but with a longer branch chain, as you see here. The next one is methionine. Methionine is unique as it has a sulfur atom in its side chain, as you see here, but still considered as non-polar due to its largely non-reactive nature. And the final one is isoleucine, another branch chain amino acid which is also non-polar. So aliphatic amino acids have side chains that is primarily made up of hydrocarbons or carbon and hydrogen, making them hydrophobic. The second class is polar uncharged amino acids. Polar uncharged amino acids, as you see in serine, there is an OH group that contains functional groups such as hydroxyl group or amide group, which can form hydrogen bonds but do not carry a charge at physiological pH. That is why it is called as uncharged, even though it is polar. The first one is the serine, which is having an OH group or a hydroxyl group in its side chain, making it polar and able to form hydrogen bonds. The second one is threonine, which is similar to serine but has an additional chiral center, here the second carbon. It is polar due to its hydroxyl group, as in serine. The third one is cysteine. Cysteine contains thiol group or SH group. It is also called a sulfhydryl group in its side chain, which is responsible for its polar properties. This group allows cysteine to participate in hydrogen bonding, although not as effective as this hydroxyl group found in other polar amino acids. So some biochemists consider cysteine as more hydrophobic and classified under non-polar amino acids. The next one is proline. Proline also has an unique structure because of its side chain that forms a ring, as you see here, with the amino group providing the rigidity. Due to its aliphatic nature, as you see, carbon and hydrogen of its side chain, proline is considered hydrophobic and non-polar by some biochemists. The next in the list is asparagine. It has an amide group. This is the amide group in its side chain, polar and can form hydrogen bonds without ionization. And the next one is glutamine. Glutamine, as you see, similar to asparagine with an additional methylene group, polar and involved in nitrogen transport in the body. So in polar uncharged amino acids have side chains or functional groups that are hydrophilic like hydroxyl group or amide group, but they are uncharged at physiological pH. That's why called as polar uncharged amino acids. The next in the list is positively charged amino acid. It's very easy as these amino acid carries a positive charge at physiological pH. Amino acid lysine contains an amino group that is positively charged at physiological pH. Then the next one is arginine. This has a guanidino group that carries a positive charge. It's a functional group with a central carbon atom that is bonded to 3 nitrogen atoms, 1, 2, 3. The next in the list is histidine that contains an imidazole ring, as you see here. It is a 5-membered heterocyclic ring containing 2 nitrogen atoms, 1 and 2. At non-adjacent positions, there is a CH in between, so these nitrogen atoms are located at non-adjacent positions. The next in the list is the negatively charged amino acids. Negatively charged amino acids carries negative charge at physiological pH. First one is aspartate or aspartic acid. It contains a carboxylate group, as you see here, that is negatively charged at physiological pH, that is involved in neurotransmission. Then the second one is glutamate or glutamic acid, similar to aspartate but with an additional methylene group, also negatively charged because of this carboxylate group, and also plays an important role in neurotransmission. And the final group is non-polar aromatic amino acids. So these amino acids, as you see here, contains aromatic rings in their side chains. These rings contribute to the hydrophobic nature of these amino acids. First one is phenylalanine, that contains a benzyl side chain, as you see here, non-polar. And the second one is tyrosine, which is similar to phenylalanine, but with a hydroxyl group attached to the benzene ring. It has some polar characteristics due to this hydroxyl group, but largely considered as non-polar due to its aromatic ring. The next one is tryptophan. Tryptophan contains this indole ring. It's non-polar. Indole ring consists of two rings or a bicyclic composition, fused bicyclic structure. This is the first ring, a 6-membered benzene ring, and this is a 5-membered pyrrole ring that contains a nitrogen atom. Let me summarize. Non-polar aliphatic amino acids possess hydrocarbon side chains. Non-polar aliphatic amino acids are glycine, alanine, valine, leucine, methionine, and isoleucine. How to remember these 20 amino acids? Based on the classification. You can watch our video on amino acid mnemonics. The link is in the description. The second class is polar uncharged R groups. The second class is polar uncharged amino acids that include serine, threonine, cysteine, proline, asparagine, and glutamine. Then positively charged amino acids are lysine, arginine, and histidine. These are positively charged at physiological pH. Then negatively charged amino acids are aspartic acid and glutamic acid because of the negative charge at physiological pH due to the presence of this carboxylate group. And finally, aromatic non-polar amino acids are phenylalanine, tyrosine, and tryptophan, all with aromatic rings. Hope you are clear. Take care, stay blessed. Thank you so much. You are with biologyexamsforyou.com.